Good morning, everybody. My name is Dr. Del Re. Today I'm going to talk about the genetics of schizophrenia. When we are going to talk about the genetics of a disease like schizophrenia, we also have to talk about a dilemma that has been around in humanity for thousands of years, I guess. What is the contribution to a disease of nature versus nurture? Let's look at this cartoon here. There is a child in the middle of the cartoon, and what you see at the left is a symbolic representation of nature, the double helix of the DNA, atoms, and cells in the central nervous system. To the right of the child, you see nurture, or the environment, the family, the community, and those tools that are available to this child. Books, an iPhone, for example, or a computer. Now, how do we know what is the contribution of genetics versus the contribution of the environment to a certain disease? A disease such as schizophrenia. In order to try to answer these questions, we are going to look briefly at the genome, at the importance of twin studies, at the importance of genome-wide association studies, and we are going to tailor these topics to schizophrenia. Before we start, we are going to try to understand whether schizophrenia is a complex or a single gene disease. In general, when we talk about diseases, we can distinguish them in single gene versus complex diseases. When a disease is traceable to a single gene, it is called single gene. It's a type of a simple disease. For example, you see here in the cartoon at the top right that this person has a problem in one chromosome. So here represented are 1, 2, 3, 4, 5, up to 23 chromosomes. The last one is the sex chromosome. You can trace the origin, the cause of cystic fibrosis, just in a gene in a single chromosome. This is not the case for all diseases. Some diseases are called complex. For example, schizophrenia is a complex disease. Let's look at the bottom part of the cartoon. This person number 2. As you can see, there are problems across all the chromosomes. And we will go a little bit more in detail about what these problems can be. Let's look at the genome. What is the genome? The genome is the complete set of DNA instructions that are found in every cell. As you can see here in the cartoon, this is a simplified representation of a cell. Inside the cell, you can see the nucleus. Inside the nucleus are those chromosomes we were just looking at. The DNA is made of 4 different chemicals. These are called nucleotides or bases. Here they are in detail. Each of these nucleotides or bases are represented by a different letter. Adenine, A, thymine, T, cytosine, C, guanine, G. You can see them here. They compose the DNA. The DNA is compacted into the chromosomes inside the nucleus of the cell. Now, the order of the letters or DNA sequence encodes the information that instructs each cell not only what to do, but also when to do it. The genome's DNA, again, is packed into the chromosomes. Now, most human cells have 2 copies of the human genome. 1 copy is inherited from the mother, 1 copy from the father, as you can see here in the cartoon. Again, we see 23 chromosomes, including the sex chromosomes, XX in the mother, XY in the father. And this person here is you or me. We have inherited 1 copy of each chromosome from each one of our parents. Remember that 1 copy of the human genome contains around 3 billion nucleotides. The 3 billion nucleotides are distributed among the 23 chromosomes. Now, how do genomes differ among people? What part of the genome makes us unique, who we are? We know that any 2 people's genomes are, on average, 99.6% identical, and they are different only at 0.4%. This difference reflects both single nucleotide differences and differences that involve multiple nucleotides. The differences among human genomes are called genomic variants. Now, if you look at the bottom here in the cartoon at the left, you see the different nucleotides or bases represented by letters. 0.4% of these letters will be different among different people. So, again, a person's set of genomic variants is part of what makes them unique. But this is not the whole story. There are other factors, as you can see here in the top cartoon in the right. The diet, the environment, the lifestyle, the social context, all of these variables are super important in contributing to a person's uniqueness. Let's look together at genomic variants. The smallest genomic variants are single nucleotide variants. They are called, for short, SNVs. Each SNV reflects a difference in a single nucleotide or letter. For a given SNV, the DNA letter at that genomic position might be a C in one person and a T in another person. A subtype of SNV is called SNP, single nucleotide polymorphism. These types of SNVs must be present in at least 1% of the population. Let's look together at this cartoon, just to understand genomic variants. In this case, you see person 1, person 2, and person 3. And as we were reviewing together, each of these persons has a part of their genome represented by the maternal heredity and part of it by the paternal heredity. Now, person 1 has a C and a T at this position in the DNA. Person 2 has a C and another C at the same position. Person 3 has a T and another T. These are called genomic variants. There is another group of small genetic variants that are important, and these are called insertions of and deletions. And you can see them here in the left part of the cartoon at the bottom of the slide. Now, insertions and deletion variants reflects extra missing DNA nucleotides in the genome. They involve fewer than 50 nucleotides. What you see here in the cartoon are chromosomes and numbers. These numbers represent the chromosome number. This is chromosome number 4. In the first example, we see a deletion of a portion of the DNA. The resulting chromosome DNA is shorter. While in duplication, the second example here, you see the duplication of a certain portion of the DNA. The resulting chromosome 4 is longer. How do scientists detect such differences in the genome? And this is a very important question. Scientists have compiled data from the genome of thousands of individuals. So they have compiled what we call a reference genome sequence, what you see here in the cartoons in the left. When you and I ask what is the difference in our genome with respect to other people, our genome is compared to the reference genome sequence. So, until now, we have looked together at how to compare our genome, yours or mine, to a standard genome in order to see whether there are genomic variants. The problem with conventional studies conducted on ordinary people is that the participants' genetic factors and environmental factors are both different from everyone else. This makes it impossible to determine the degree of influence of the environmental factors on the disease, no matter how many people are studied. We turn then to another type of study. This study involves twins. And as you can see here, we have in the cartoon represented identical twins and fraternal twins. Because the genes of monozygotic twins or identical twins are identical, it is possible in a situation where one twin has a disease and the other does not, to narrow down the reason of the development of that disease to environmental factors. Furthermore, by additionally comparing monozygotic twins, identical twins, to fraternal twins, dizygotic twins, who share half of their genes in the comparison, the respective degrees of influence of genetic and environmental factors on the disease can be determined. Now, studies that have been based on analysis of twins have shown that the genetic contribution to developing schizophrenia is moderately high. And in addition, that the heritability is substantial, 81% roughly, by meta-analysis of twin studies. In addition to twin studies, we can try to understand the contribution of nature versus nurture in the development of schizophrenia by using genome-wide association studies, or GWAS. A genome-wide association study is a research approach used to identify genomic variants that are statistically associated with a risk for a disease or a particular trait. The method involves surveying the genomes of many people, looking for genomic variants that occur more frequently in those with a specific disease or trait compared to those without the disease or trait. For example, people that have schizophrenia and people that are not affected by schizophrenia. Once such genomic variants are identified, they are typically used to search for nearby variants that contribute directly to the disease or trait. GWAS estimates that common SNVs explain around 23% of the variants in schizophrenia risk. If we look at this cartoon here at the left of the slide, we can see how genome-wide association studies are carried out. We have a large number of cases with the disease, for example, schizophrenia, and we have a large number of controls. Controls means they don't have the specific disease we are interested in. The DNA from each group of participants is sequenced. The GWAS will compare the variants in the genome of the cases with disease to those of the controls. When we look in particular at schizophrenia, a study in 2014, a very important study published in 2014, demonstrated that there are 108 common variants that are significantly associated with schizophrenia. The study was carried out by GWAS, Genome-Wide Association Study. Now, those variants, are they important? First of all, we can look at this image, this picture from the paper from 2014 and try to understand it. What we see here on the x-axis, the horizontal axis, is the number that represents each chromosome, 1, 2, 3, 4, 5, up to the sex chromosomes. What we see on the y-axis is the significance of the association with the disease, schizophrenia. This red line here represents the significance limit for these associations, meaning that anything above this line is significant. The variants, as you can see in the genomes, are distributed across the different chromosomes. And this reminds you that we are talking of a complex disease where there is not one gene explaining the disease, but many variants across the genomes that significantly associate with schizophrenia. What do these variants represent? 75% of the variants include protein coding genes. 8% are within 20 kilobases of a gene, meaning they are close to a gene. Among the variants, there are important factors that we know are essential in the central nervous system. DRD2, for example, the receptor for dopamine, an important neurotransmitter affected in schizophrenia. Among the variants, there are also glutamatergic neurotransmission important genes. These are named here, GRI1A, GRIN2A. And then there are variants that are portions of genes that are important for calcium signaling. And remember that calcium is important in synaptic transmission, the communication between different neurons. We have just seen together that the genomic variants that are associated with schizophrenia are distributed across the genome. So, how does the collection of a person's variants affect the risk for schizophrenia or for other diseases. Researchers identify genomic variants associated with complex diseases by comparing the genomes of individuals with and without that disease to calculate the variants that are more frequently associated, that are more frequently found in people with that disease. Nonetheless, there can be hundreds or even thousands of variants per disease. The way we use this information is to calculate a polygenic risk score. A polygenic risk score is the sum of all these genomic variants that altogether increase the risk of developing a disease or that are associated with the disease. Then what we can assume is that a polygenic risk score explains a relative risk for a disease. Indeed, the data used for generating the polygenic risk score comes from large-scale genomic studies. These studies find genomic variants by comparing groups with a certain disease to a group without the disease. That is, they do not provide a baseline or a time frame for the progression of the disease. So again, let's look a little bit more in detail at the polygenic risk score. If you look at the left side of the cartoon here, you see the distribution of the polygenic risk score in controls in black and in cases in red. On the x-axis, what you see is the distribution of people across standardized polygenic risk score. The controls have a distribution that is more to the left than cases. That means that cases have a higher number of variants that are associated with the disease. In the right side of the cartoon, what you see is the polygenic risk score with respect to age. So on the x-axis, you see age from 0 to 80 years old and different types of polygenic risk score. In red is high polygenic risk score and in blue is low polygenic risk score. Meaning that if you look here at the cartoon to the left, the left portion of the slides, the low PRS will be here in the middle, while the high PRS will be on the right side, this tail here of the distribution in the population of risk variants. The high PRS here, represented in the right part of the cartoon, shows that when the PRS, the polygenic risk score, is high, it increases for predictive ability with age. This should be clear if we think about, for example, coronary artery disease. Young people can have a high polygenic risk score for coronary artery disease, but at the same time, coronary artery disease is a disease of older ages, and so the predictive ability for coronary artery disease for the PRS will be higher for people that are older than for people that are younger. When we look specifically at schizophrenia, the polygenic variation contributes to impaired brain function in schizophrenia for 23% of risk to developing schizophrenia. So the common variants that altogether constitute the polygenic risk score explain 23% of the risk in developing schizophrenia or having schizophrenia. Schizophrenia is a complex disease, and we see that the common variants explain 23% of the risk. So, twin studies show us that the heritability is 80%, meaning that scientists are puzzled by what is called missing heritability. The common variants in the genomes explain 23%, but twin studies and also family studies show that the heritability, the genetic contribution to the disease, to schizophrenia, is much higher. There must be something else that explains why schizophrenia and other diseases are so heritable, meaning why they run in families. If you look at this cartoon, the heritability is represented on the y-axis, this vertical axis, and 0.8 means 80% heritability. The true contribution of genetic factors is represented by this darker first column, while the overestimated heritability by this lighter portion of the column. We know by looking at the common variants and looking at the second column that common variants explain around 25% of the genetic contribution. Other genetic variants, genomic variants, can be important, but they also explain just a little bit more of the heritability. This means that most probably the environment, represented by gene-environmental interactions and other modification of the DNA that is called epigenetics and that find their cause in the environment, play a very important part in the puzzling missing heritability of schizophrenia and other mental diseases. Now, if we look at this cartoon from a paper published in 2019, we can try to understand what are the contributions of the environment and the genetics to schizophrenia. On the x-axis, we see age. It starts with a -1. -1 because we are talking about the influence of genetics and the environment to the baby in uterus. 0 represents the birth of the child, and then we see the age increasing. Notice that around age 20 there is this word onset, because the majority of people that develop schizophrenia develop the disease around age 20, in the late teens and early adulthood. Now, let's look at the cartoon in detail. When we look at early age in the neurodevelopmental trajectory in the life of a person that has schizophrenia already in early stage of their life show subtle motor, cognitive, and social deficits. This might be due to SNPs and variation in developmental genes. But also, as you can see here in purple, prenatal and perinatal events. The prenatal and perinatal events include environmental factors such as infections when the baby is still in uterus, poor diet for the mom, stressful events for the mom that are passed to the baby through epigenetic factors, methylation of the DNA, poor diet, and also, for example, drug use. These are environmental factors. After birth and in childhood, there might be other problems that come around in the life of this child. Environmental factors are represented by chronic social adversity during childhood, migration, urbanicity, poverty. It is here that an interaction between genetics and environment occurs. So the child and the young teen will manifest problems such as social anxiety, quasi-psychotic ideas, and depression. And remember that chronic social adversity in childhood causes hyperactivity of the HPA axis, meaning the axis that regulates stress in the body and also inflammation. This young person is growing. The social anxiety and the quasi-psychotic ideas with the contribution of the stress body reaction and increased inflammation will have dopamine dysregulation, for example, so dysregulation of neurotransmission. Here, genes that contained variants that are associated with schizophrenia in those neurotransmitters or receptors for those neurotransmitters will be involved. At the same time, there are other environmental factors that can come into play. Drug abuse, for example, cannabis, use of psychostimulants, smoking. These environmental factors also by themselves have an impact on glutamatergic neurotransmission. Glutamate is an important neurotransmitter in the central nervous system, and they will also impact the function of the endocannabinoids in our system. All of these factors together are linked to development of schizophrenia. And so in this cartoon, what you see is really a summary of the interaction of genetics, represented here by a G, and the environmental factors, represented here by a E. We are going to conclude this lecture by reiterating that schizophrenia is a complex and polygenic disease. It's a complex disease, meaning that is not due to a problem in a single gene, such as, for example, cystic fibrosis, and is a polygenic disease because it's the result of many variants in the genome that together increase the risk of developing schizophrenia. But most importantly, the risk contribution to schizophrenia is both from the environment and from these genetic variations that we have been talking about In fact, when we look at irritability, genetic variation explains maybe a little bit more than 20% of the irritability, while twin studies we saw explain around 80% of the irritability, suggesting that there is what we call a missing irritability of schizophrenia. Most probably and most logically, the other big contribution to developing schizophrenia comes from the environment. So we can rationally say that the etiology or cause of schizophrenia is multifactorial and reflects an interaction between genetic vulnerability plus environmental contributors. Environmental risk factors such as pregnancy, birth complications, childhood trauma, migration, social isolation, urbanicity, substance abuse, alone and in combination, acting at several levels over time, influence the individual's likelihood to develop the disorder. I want to give a message here. Many of these environmental contributors are preventable, and it is our responsibility to prevent those. Poverty, health care, access for everybody, the isolation of people, the use of drugs, aggressive environments, all of these are contributing factors to mental diseases that are preventable. Thank you so very much for listening to this lecture. To the next one. Goodbye.